All right, guys. So far we have been talking about talked about viruses. Talked a little bit about phylogenetic trees. Last time we started talking about this branch of the phylogenetic tree, the eubacteria. Today we're going to continue that. We're going to talk also about this branch, the archaebacteria, and when time permits, we'll talk about a couple of these early branches on this tree as well, some of these guys. Essentially in this first set of study sessions, bacteria, archaea, and protists is kind of where we're trying to go with this. But
we need to start with inch sheet.
It's coming.
Okay.
Where the sign-in sheet make it to, guys. So yeah, bring it back around this way when you guys finish. So I'm going to pick back up with bacteria today. We spent a lot of time on viruses already. Eubacteria is the first group that we mentioned. We know they have prokaryotic cells. No enclosed nucleus, no membrane-bound organelles. Remember they have a cell wall. It's made out of a kind of a unique material called peptidoglycan. Part protein, part what? Part sugar. Remember that the eubacteria, even though they don't have membrane-bound organelles like mitochondria or chloroplast or ER, they do have ribosomes. Ribosomes are essential to all living things because what do? They make proteins. And what's no good if you don't have ribosomes? Your DNA is no good, right? Because that's what DNA does. It codes for proteins. So every living thing has to have ribosomes. The ribosomes in bacteria are a little bit different in structure than the ones in eukaryotes, but they have the same function. Remember that bacteria do have DNA, but not a whole lot. They have that single circular chromosome, just kind of floating around out there in the center of the cell. Sometimes we call it the nucleoid. What do we say that word literally means, nucleoid? Yeah, kind of like a nucleus. It's not a nucleus, but it's sort of like one. Also remember that some bacteria on the outside of their cell wall have this mucus-like coating. You might see it called the glycocalyx, because it's part sugar. Or you might see it called the capsule. And when we were talking about this last time, what did we say it does? What's its function? It protects from immune systems. It could also protect from dehydration or desiccation, drying out. It can help them move because it's kind of slick. Another thing we talked about is how bacteria reproduce. We said usually, this is the way they typically reproduce. Prokaryotic fission, or a lot of you know it better as what? Binary fission. It's just when one splits into two. It's kind of like what kind of process that happens in you. Mitosis. It's kind of like mitosis. It's cloning. It's asexual. And we've said over and over again, that's a great strategy as long as what's the case? Steady environment that you're good at as an individual. Asexual reproduction makes a lot of sense here. But it doesn't make much sense when what's going on? When the environment's changing and you're not changing with it. So bacteria have also evolved a almost sexual-like reproductive strategy, and it's called conjugation. And that's when two bacteria get up near each other, and they attach these special structures called pili. My green lines are going to represent a single pilus. It's like a tube almost. And that hooks them up. Now remember that each one of them has this loopy chromosome in the middle. They can't really do much of that because that's their one chromosome. But what do they have in addition to that one main chromosome? Yeah, they got these little bitty loops of DNA. Really, really tiny loops of DNA. And those are called plasmids. And during this process that we're looking at here of conjugation, they attach, and they can switch those back and forth. They can swap them out. And that's sort of what sexual reproduction is, right? Sexual reproduction is about mixing genes from two different parents. Even though when these things split, there's still just two bacteria, the bacteria are different than they started with, right? So it's still a form of sexual reproduction. Even though it doesn't make any extra organisms, it makes new organisms, new versions at least of organisms. All right, another thing we talked about were the different shapes bacteria tend to come in. What's the sphere-shaped one called? Coccus. What about the one that's shaped sort of like a capsule, like a medicine capsule? It's called a bacillus. Sometimes it's called a rod shape. I like to think of it more like a capsule. And then there's also the little corkscrew shape, which is called a spiral. Something that I don't think we talked about last time was this thing right here, endospore. Let's talk about that one a little bit. Let's say that you're a bacteria, you got your little piece of chromosome here, and that you're living in an environment that has become kind of nasty. Maybe it's gotten really cold, or really hot, or maybe it's acidic, or maybe it's salty. Something weird is going in the environment. You can't handle that environment. You're going to die as a cell. Some bacteria have the special ability that when the environment starts to get really bad, they make this thick coat around their chromosome. Well, what happens to the cell? The cell dies. But that thick coat around that DNA is very resistant to extreme environments. So once the environment gets better, essentially that DNA just what? It makes a new cell. But it's exactly the same as the one that was there before. This makes some bacteria pretty hard to kill. Because you're trying to kill them and you think they're dead, but they're really not. They're just sort of hiding, waiting for things to get better. Those are endospores. We talked a little bit last time about some bacterial diseases as well. There are a lot of bacterial diseases out there, everything from strep, tetanus, better known to some people as what? Lockjaw, syphilis, the STD, pneumonia. We talked about tuberculosis or TB. Black Death was bacterial, right? Is the Black Death still around? Yeah, people get it. There are a few cases of it every year. It's just now, why is it not such a huge deal? It's treatable with penicillin. Now, I still wouldn't want to get the Black Death because when do you think the last time your doctor saw a case of the Black Death was? Never. And the thing is, if you don't figure out what it is and you don't start treatment pretty early, it's not treatable anymore. So the first few cases that showed up in an area, those people might die because their doctors might not know what it was. They might think you got something else going on, and it might take a while for them to figure out what it is until they do some blood work and those kinds of things. And by that point in time, it could be too late. We realize that most bacterial diseases are treatable with antibiotics. That's what antibiotics are all about. And when we were talking about enzymes and such, how do we say most antibiotics work? They're inhibitors. They inhibit enzymes that bacteria have that we what? That we don't. So we end up killing them off, or at least we keep them from reproducing. But it doesn't have any effect on us because it's inhibiting enzymes that we just don't have a copy of. We mentioned the bacterial diseases, but remember kind of where we left off last time. Bacteria are really way more positive than they are negative. All kinds of positive things they do in the environment. Maybe the most important is decomposition, breaking down stuff. Old leaves, waste, dead bodies. We talked about how they essentially cause the nitrogen cycle to happen. When you take N2 out of the air and convert it to either nitrate or ammonium ion, that's called what? Which one of those two words? That's fixation. And that's carried out by bacteria that are living on tree roots sometimes, maybe on the roots of soybeans, maybe just in the soil, maybe in the water, like in the oceans. But they turn unusable into something that's usable for everything, ammonium or nitrate. And there are other bacteria that do just the opposite, where they take those usable forms of nitrogen, ammonium, nitrate, and convert them back and put them back into the air as N2. And that's sort of how nitrogen is recycled over and over again in our environment. We talked a little bit last time, too, about this idea of bioremediation. How that some scientists are starting to use bacteria to do what? Clean up certain kinds of environmental issues like oil spills. They've actually found bacteria that can essentially digest oil to help to clean up that kind of mess. And then I think the very last thing we talked about in the last session were these three words at the bottom. What's it mean to be an aerobe? Use oxygen for cell respiration. Some bacteria are aerobic, just like you are. They have to have oxygen to be able to carry out very much cell respiration. Some bacteria, on the other hand, are obligate anaerobes, which means they have to be in a place where there's not much oxygen. They don't deal well with oxygen. And then there are still other bacteria that are sort of like the switch hitters of bacterial world. They're called facultative anaerobes. Essentially, they can deal with oxygen or they can deal without. So bacteria, there's a wide range of different ways they approach cell respiration. Some approach it just like you do, aerobically. Some approach it pretty much completely anaerobically. And some can do a mixture of both. Any questions here? There's a picture of a typical bacterial cell. Some bacteria have flagella to help them move. A lot of bacteria have these little hair-like things on them. These aren't cilia. Those are the pili we were talking about earlier. We said that some of those pili are specialized to help them swap plasmids, but other pili are there to help them do what? Not so much move, attach. Think of them like Velcro. Help them attach and hold on to surfaces. Also notice the capsule on the outside, that's that mucus coating. And then underneath that's the wall. What do we say one of the main purposes of a wall is for a cell? Protection from what? Water. From not it can't keep the water out, but it keeps the cell from what? Bursting, because it helps to hold up the pressure. All cells have a membrane, that's in blue in here. Pretty much the same phospholipid bilayer your cells have. Notice there's little ribosomes that are freely floating all throughout the cytoplasm. This is the main chromosome. But remember, that's a lot of bacteria. Most bacteria have little extra loops of plasmids. Those are the ones that they swap during that sexual form of conjugation. All right, this is just showing you binary fission. How that the bacteria starts with one loop, it copies it. Those loops are attached to the membrane. The cell grows a little bit, and eventually it just splits in half. That can happen in as little time as 20 to 30 minutes for some bacteria. In the perfect world for bacteria, one bacterium can become several million in a 24-hour period. And that's kind of scary. This is showing you the different shapes. Here are spheres in a chain. This is what we call streptococcus. Except that causes strep throat and other disorders. Strepto means a chain. So it's a chain of spheres, streptococcus. Here's some bacilli, the kind of capsule shape, like a medicine capsule. And then this one's hard to see because it's in this dark color. But it's sort of a spiral shape, a spirillum. Another thing here, there are some bacteria that can do photosynthesis. And the main kinds of those are cyanobacteria. A lot of books and a lot of people used to call them blue, green, what? What they used to be called, blue, green, what? Algae. They used to call them blue, green algae. Why aren't they algae? Why is that wrong to call them blue-green algae? Well, algae have what kind of cells? Eukaryotic. True nucleus, chloroplast, mitochondria. These don't. They're prokaryotic. They've got a freely floating piece of DNA. Now, for a plant to do photosynthesis, it's got to have chlorophyll, right? Do these things have chlorophyll? Yep. Where's the chlorophyll at in a plant? in the leaves, but zoom on in. Go from leaf, keep zooming. Cells, keep zooming. Into the chloroplast, keep zooming. Yeah, there's these little membranes inside the chloroplast called thylakoids. And these chlorophyll's a protein. And it's embedded, it's like stuck in the cell membrane in a certain order. Well, these things don't have chloroplast. What's the only membrane they have? Yeah, the cell membrane, plasma membrane. So all their chlorophyll and all the things involved in photosynthesis are essentially embedded in their cell membrane instead of in the little membranes of the thylakoids. Similar process other than that. Where it happens is different, but how it happens, very similar. All right. What we'll talk about for most of the rest of our time today are these guys, archaea. And remember, a lot of people look at that word and think what? Oh. And originally, scientists named them this because what did they think? They thought they were the oldest things on Earth. But did that phylogenetic tree we looked at a minute ago back that up? It didn't. Since they've been studying them, scientists have essentially decided that they're in between the bacteria and the eukaryotes. And that they actually, right in here, they share a common ancestor with only eukaryotes. Here they share a common ancestor with bacteria and eukaryotes. But remember, on these phylogenetic trees, as you go this way, what's implied? It's older in terms of evolution. So eubacteria, according to this, came before the archaea bacteria. Oops. Going the wrong way there. Now, the way the archaea are talked about in the textbooks you probably use for regular biology is they talked about them always being in harsh environments, what they call extreme environments. Like really hot, really cold, really dry, really salty, what they call extreme environments. But what have they since found? These are really everywhere. There are some of them that can live in really extreme places. But there are some of them that just live in normal, healthy environments. What they found, though, for a long time, these guys and eubacteria were classified together. They were classified into a kingdom called Monera. But what do we know our classification system is built on today? Genetics and evolution. And what they've discovered as they've been studying these things more and more is they're pretty distinct from regular eubacteria. They're genetically different. They evolved in a different pattern in different places. And so now they're classified as their own group. Not this kingdom Monera doesn't exist anymore, done away with. I've got a whole slide that goes through some of their differences, but here's just a short list. The cell walls. Just like bacteria, they have cell wall. It's made of completely different stuff. It doesn't have, what was that material in the regular bacteria cell walls? Peptidoglycan. Not found in these guys. Completely different material. Their cell membranes are made in a slightly different way. The RNA that's used to make up ribosomes is different. It's actually more similar to that in a eukaryote than in a bacteria. And we'll get more details of that in a minute. But remember, some of them can survive in really extreme places, like volcanic vents, hot springs, like you might find in Yellowstone or in other places around the world. Really highly salty water, like say the Dead Sea or the Great Salt Lake. And the extremophiles, the ones that live in extreme places, are usually classified into three groups. They're the methanogens. What's methane?
CH4.
Yeah, methane, CH4, natural gas. These things actually usually live in like swamps, and they can produce methane. Then there's a group called the thermoacidophiles. What's file mean? Yeah, they like love it. So they love hot and acidic places. Thermoacidophiles. And then there's also the ones that are called extreme halophiles. What's halophile mean? Like halogen, for example. What's the word halogen mean? In chemistry. The group 7 elements. Why are they called halogens? It means they form salts, salt formers. So these guys love salt. Extreme halophiles, salt lovers. We'll come back to some of this stuff in a little bit. There's just some pictures of some archaea. Notice they have some really unique shapes. This wouldn't fall into their shapes we talked about a minute ago. They can be found in these hot, salty, or acidic springs. This slide may be more important than the last one, though. This slide sort of compares the similarities and differences between the eubacteria and the archaea. And let's look at the similarities first. Both have prokaryotic cells. No membrane-bound nucleus, no membrane-bound organelles. They both have cell walls. And in terms of their size and their shapes, somewhat similar. Both way smaller than what? Yeah, your cells. Their cells are way smaller than your normal eukaryotic cell. And they tend to reproduce in the same basic ways, binary fission usually. But over here is the differences column. We said they both have cell walls, but the archaea, their cell walls are not made out of peptidoglycan. They're usually made out of lipids. Cell walls are made out of these unique lipids.
Not like.
Yeah, a different kind of lipid, but still kind of maybe more wax-like. Their ribosomes and their rRNA seem to be way more like ours than they are bacteria's. And I'll also throw in another word in this group. Anybody know what that is? A histone?
It's what DNA wraps around like a little ball.
Yep. There's these little proteins in your cells that when the cell's about to do mitosis or meiosis or divide. Now, well, let me backtrack. What does your DNA normally look like? Just a normal, everyday cell doing its thing. Its DNA is sort of like a string that's unwound all over the place. Spaghetti is a pretty good analogy. If you had, how many chromosomes are in the normal human cell? 46. Think if you had 46 spools of yarn. And what's going to happen before cell division anyway? Before that. What's going to happen to those 46 spools before that? It's got to get copied, right? So really there's 92 spools of thread, right? Let's say that I walked into the room and I had 96 spools of yarn, and I told Adam to stand over the door and hold one end of the yarn, and then I threw them across the room. And then I came over to Sam and I said, Sam, you've got 2 minutes to separate these 92 spools of thread into 2 exact sets. Is that probably going to happen?
No.
Probably. Sam's good. He's fast. But he's not that good or that fast. When's the only time your DNA is in the form of a chromosome? Just before, during, and just after what? Mitosis. Or meiosis. Because what's the purpose of a chromosome? It's like a spool. It's a whole lot easier to separate 92 spools of yarn than it is 92 unspooled spools of yarn, right? That's the purpose of a chromosome, to wind it up, make sure that each new cell gets 46 exact chromosomes. The histones are the proteins that are sort of like spools. What the DNA winds around to form those chromosomes. And what I'm saying up here about the archaea, those proteins in them are a lot more like our eukaryotic histones. And actually, regular bacteria don't have them in the first place. We have histones as eukaryotes. Archaea have them. Bacteria don't. And since they're proteins, what else does that mean we share? Where proteins come from? Which, what else has to be involved? DNA, right? Because there has to be some directions for building those proteins. Which means we share some genes with archaea that we don't share with those other bacteria, the eubacteria. Another difference, some of the eubacteria can form those endospores, those protective coats when the environment gets bad. As far as we know, archaea don't do that. Also, as far as we know, there have been no archaea that I'm aware of found that made people sick. All the pathogenic bacteria are the eubacteria. So again, it's the proteins that your DNA winds around when it's making a chromosome. Eukaryotes and archaea have those. Regular eubacteria do not. That makes sense? All right, we're going to completely change gears then. And we will start our other topic. It's going to be a little bit different setup here than you're used to. You guys familiar with Prezi? Yeah, this is another piece of, this is an online piece of presentation software. It's kind of like PowerPoint, but it'll do this cool zooming thing. It's free. Yeah, it's free.
No.
Oh, I'll make it available to you. Yeah, I'll make it available to you. I'll put a link on Edmodo for this. All right, but we're going to talk about protists. Now realize, first of all, that word protist is actually probably not a correct word. What did we say they decided about protists?
It's not.
Yeah, it's not really a kingdom anymore because what they find out about them is they were studying.
No.
They're really, it's just a big group of things that were just stuck together because they were mostly white. What made a protist be a protist?
Make it classified as.
Yeah, but what did the things they classified as protists, what did they have in common usually?
They just didn't else.
Yeah, but what were some things they did have in common though? Mostly they were single-celled eukaryotes. So they just threw them all together because they were single-celled eukaryotes. But when they started looking at their DNA, what did they find? It's really different. So this is just a kind of a human made-up kingdom that probably really shouldn't have ever been a kingdom. But we still don't know where to put them exactly, so we still talk about protists a lot because they don't really fit anywhere. Yeah, here's a phylogenetic tree. What does, well, right in here, what would be implied to be there? Common ancestor of what?
All that stuff.
Yeah, probably literally right here would be between archaea and protist, right?
Mm-hmm.
But what does this imply? Yeah, there's some protist-like thing that was the common ancestor of plants, animals, and fungi, right? That all the multicellular eukaryotes evolved from some kind of prokaryotic protist-like thing. Again, another phylogenetic tree. Let's get this writing out of the way. So the protists, at least the things that we used to call protists, are eukaryotic, as we said before, mostly unicellular. Well, there are a few things, like certain kinds of algae, that are sometimes multicellular. Some things that we call protists are kind of animal-like. They were heterotrophs, had to eat. But there were also a lot of things that were more plant-like and were autotrophic, could be photosynthesis. And then that last sentence, you may not know what I mean by that. All are complex generalists. What does that mean? It means the one cell had to do what?
Everything.
That one cell had to do everything. It's complicated. The cells are way more complicated than bacterial cells because they have organelles, but it's still a one cell has to be able to do everything kind of setup. As I said a minute ago, they're thought to be the first eukaryotic organisms. That's what came before protists. According to that phylogenetic tree we were looking at a minute ago, what came before them? Archaea and on back, which was eubacteria, all right? But the first cells with organelles and a membrane-bound nucleus are thought to be something protist-like. And also, as we said from that diagram, therefore, they're probably the descendants of all plants, all animals, all fungi. Now, we talked several times about the endosymbiotic hypothesis. We haven't talked so much about this idea of membrane invagination. We'll talk about both of those here in a second. What's the, actually, let me see what's on my next. Let's look at this one. This is the idea of endosymbiosis, or the endosymbiotic hypothesis. And the idea there is that a big prokaryote did what? Took in some smaller ones. Probably if bacteria could have intentions, what was the intention? To eat it. But for some reason or other, it didn't ever digest it. And it turned out that together, they were a pretty good team. And that some of those that got eaten, for lack of a better word, might have had the ability to do what? Like, maybe what functions? Yeah, maybe like make ATP. Maybe like make sugar, photosynthesis. Maybe move in a weird way, like a flagella or something. So the idea is that a large prokaryote started taking in smaller ones, and that it turned out to be a good mutualistic relationship, and that over time those eaten ones became what? Organelles. We talked about several pieces of evidence for this, because when the lady who came up with this idea first stated it, her late name was Lynn Margulis. Most of the scientists sort of laughed at it. That's the stupidest thing I've ever heard. But then they started looking, at different organelles and such. For example, like the mitochondria. And what did they find about mitochondria and chloroplast that might make them think maybe she's not so wrong after all? Yeah, they got their own DNA. And what does that DNA look like? In a mitochondria and a chloroplast. It's a single what? Single, what kind of strand? Single circular strand of DNA. Just like what? Just like bacteria. They have their own ribosomes. And the ribosomes in a mitochondria and a chloroplast are a lot more like what than what? They're a lot more like bacterial ribosomes than they are the ribosomes found in your normal cells, than the eukaryotic ribosomes. They also can essentially do binary fission. The mitochondria in you can reproduce independently of the cell that they're inside. They can reproduce on their own. Chloroplasts do the same thing. Not to mention the most obvious ones is that they're about the same size and about the same shape as your typical bacteria. So all things that Margulis and other people use to back up this idea that maybe eukaryotic cells evolve through this process of endosymbiosis. All right, let me get the writing out of the way. All right, I'm going to go past this one for right now. Like we said before, Protista is usually not considered a kingdom anymore. The members are put into either other kingdoms, like, for example, some of the algae used to be protists. Now they're classified as what? Plants. But most of them are just sort of put into what they call non-conforming clades, which essentially means what? They're in a group. That group doesn't seem to go with anything else. We're not going to call it a kingdom because there's not enough things in it. But they're just in this group that's out there in la la land, and we don't really know where to put them. Okay? That's what we mean by a non-conforming clade. Maybe we'll figure it out one day. Maybe not. All right, and probably where we'll end today is how do protists get food? How do they get their nutrition? Turns out there's different kinds of protists. There are protists that are heterotrophs, which makes them sort of like us animals. They have to eat. What does the word protozoan actually mean? What's proto mean? Like a prototype, for example. It means like first. What's zo mean? First animal is literally what protozoan means. Now, are protozoans animals? No, but they're kind of animal-like, right? They eat. They also usually what? It would be kind of animal-like. Move, reproduce. We're talking about things here like parameciums and amoebas. They have to eat. They can move, but they're one-celled, what we used to call protists. There are also a lot of protists that look a lot like fungi. There are these things called slime molds. It's gooey, slimy stuff that grows on plants mostly. Both of those have to eat. They break down food. The protozoans usually do what we call intracellular digestion. What does that mean? Intracellular digestion. They what do they do to the food before they digest it? They pull it in, right? They like phagocytize it. They pull it in, and then what organelle helps them to digest it? A little lysosome helps them to digest it. But what now? Yeah, that's intracellular. The protozoans. But a lot of the fungus-like things do what's called extracellular digestion. Like bread mold, for example. Bread mold is not a protist, but what does it do to the bread or that stuff that grows on oranges, that orange mold? How does it digest it? It essentially secretes enzymes out of the cell onto the food. It's almost like it spits on it, okay? It's almost like it spits on the food. What happens to the food? It gets digested outside the cell, and then what does the cell do? Slurps it in, essentially, right? So it digests it before, and then it pulls it in. Sort of like a way of starfish. You guys know how a starfish eats? Starfish is pretty cool. Let's say a starfish is trying to eat a clam, and that's one of the things they eat the most of. They use those legs to pull on the clamshell until what gives out. The muscles that let the clam open and close its shell. And then once they get that shell open a little bit, the starfish basically spits his stomach out of his belly button. he turns it inside out and essentially sticks its stomach out into the clamshell. What does it dump into the clamshell along with its stomach? All its stomach acids and enzymes. It dumps into the clamshell. So the clam's getting digested in its own shell, and then it pulls its belly back in, and as it's pulling it back in, it's sucking in the digested clam. That's sort of that's extracellular digestion. Yeah. I don't know what time. I don't know what time for me. That's a good question. Don't know. No idea. All right. There are though also protists rather that are called photoautotrophs, which essentially means they do what? They do photosynthesis. So they're sort of plant-like. There's this cool protist we'll talk about. If we don't get there today, we'll get there next time, called the euglena. You guys have heard of euglenas before, right? A euglena is really awesome because it's really both of these. It's a heterotroph and a photoautotroph. As long as there's light, what's the euglena do? He does photosynthesis. He acts like a plant. But when the lights go out for an extended amount of time, what's the euglena start doing? It starts eating. It eats other protists, bacteria. So it's kind of like a missing link almost between plants and animals. It can act like a plant. It can act like an animal. Pretty awesome thing. Like I said, it eats other protists or bacteria, things like that. We'll get there. The photoautotrophs are things like algae. They're also phytoplankton. Let's talk about that word for a second. What's phytoplankton mean? If you ever see in biology, P-H-Y-T. A lot of times there's an E there too. P-H-Y-T-E. Fight. What's fight mean in science terminology? It means like a plant. And what does plankton mean? What are plankton? Everybody in here that throws that word around like you know what it is, but you probably don't. Okay?
They're very little dudes. They just kind of float.
Adam said the key word. There are things that live in a body of water that tend to float on the surface. So this literally means plant-like things that float on the surface. Literally what it means. Okay? Plant-like things that float on the surface of a body of water. Phytoplankton. Phytoplankton are actually where most oxygen in the world is. That's where it comes from. Most photosynthesis doesn't happen out in the rainforest. Most photosynthesis happens in the ocean. I'm not saying go cut down the rainforest. That's what I'm not implying that. I'm just saying that we better take care of the oceans, because if the oceans go south on us, then we're in trouble. How could what? Go south. I mean, bad. Go bad. Go bad. There are also some protists that are called mixotrophs, and this is where the euglena actually fits in. The euglena is both a heterotroph and an autotroph. Sort of like a switch hitter of the protist world. It can switch between being photosynthetic and between being heterotrophic. I think the next picture is probably a picture of the euglena. Yeah. So notice it's got all kinds of chloroplast in there. All those green objects are chloroplast. Normally it's going to do photosynthesis. Notice it's got a long little flagellum that helps it to move. It's got a little eye spot. A little red dot right in here. Now, can it see? Probably not. What's that eye spot all about? It's a light detector. If you're a euglena, what's your light detector going to tell you? There's light over there, so what are you going to do? You're going to go there, right? Because if you can go there, you can make food. So it's just to tell it to move to the light. All right, we'll mention this and then we'll call it. Most protists are motile. What does it mean to be motile?
Can
Can move, capable of movement. And there's three main ways that protists move. They move with flagella, cilia, or pseudopods. What's a pseudopod?
it's like a little leg.
It means fake foot. What's the famous protist that moves that way?
Okay.
Yeah, little amoebas. It's almost like they're. You guys ever seen the movie The Blob? It's like an old horror movie. This blob just sort of flows after you, right? And it flows around you and engulfs you and it kills you. That's kind of what amoebas do. And I'll actually, I think I'll save this video for the next one of these. But I have a video, one or two more frames in. It shows an amoeba flowing around the paramecium as it eats it and digests it. It's pretty cool to watch. We'll stop there. Let me get this video stopped right quick.
Are you going to put the other power on there too?
I can.